We talked before about the structure of the DNA molecule and how it relies on a lot of proteins for its scaffolding. In terms of DNA replication, even more proteins are going to be important. And we're actually going to be describing in this quick video how DNA copies itself and what we have learned about it and how we have learned about it. So, there were three theories leading up to the actual DNA copy process. Watson and Crick thought that it was kind of like the semi-conservative model, but they actually hadn't come up with a way to prove it. It was two scientists called Mesenson and Stahl which actually came up with a way to prove it. But the three leading theories were that each DNA copy was made based on the original template, but that the new copies were basically brand new. They had none of the old stuff in them. The semi-conservative model was the idea that the DNA was made of a template, but that each copy was basically half old, half new. And the dispersed idea was that the copies had intermittent copies of old and new. And all depended on how DNA actually copied itself up. Now, the answer was not actually achieved until finally the experiment was done. But think about what would happen if the DNA was conservatively copied. That means that after every so many cell generations, you will eventually get brand new material every time. It will be very hard to find out if this new copy had any defects on it since you lost the template. If you have the semi-conservative model, you can compare each new copy straight away with the template and make sure that it's supposed to be the way it is. That has obviously an advantage of the conservative model, and that's why life chose that. Even though the conservative model will make you have always new materials, which over time would actually be better for the cell to maintain its integrity, it will actually be worse for integrity because the new materials might carry more mutations with it. And so the cell actually preserves that. Besides, as you will see soon, this process still makes a lot more new material than old material over time. So basically the idea that Mesenson and Stahl came up with is that they would tag the DNA of the original cell and by making it grow into a radioactive nitrogen that was nitrogen 15. And this nitrogen 15 would demonstrate the original composition of the DNA. So they'll let the cells grow in that environment for a while. And they would drag the cells and switch them to an environment that had a lighter nitrogen tagged with a different color, nitrogen 14. Now, what they actually noticed is that after several generations, less and less of nitrogen 15 was around in the DNA, and more of nitrogen 14 was around, but that the nitrogen 15 was still present even at the very end. And so what this told them is that the DNA process was semi-conservative, as you see in the top. The way they actually did it is that they realized that by centrifuging the DNA that was made of the heavy nitrogen 15, the DNA was heavier, so they would basically fall to the bottom of the vial. But if the DNA was half old, half new, they would get a little lighter and it would be halfway in the vial. And if it was completely new, it will be completely light and it will be more towards the top. And so when they go back to the actual experiment and after many generations, they realize that after one generation, all of the DNA seems to be in the middle. That means there isn't any DNA that's made only of old types. That throws out the conservative model out the window and really proves that there's a semi-conservative model. But if that's true, in another generation, it will split up again, and you will have some strands which are basically just new. And that's exactly what they found after another generation. Some of it were hybrids, but some of it was just new. And as they did more and more new strands, pure new strands will show up, and less new strands will be hybridized. But never you would find strands which were just old after the first generation or the parent generation. And that actually proved that DNA was copied in a semi-conservative process. And I do talk about this in more detail in another video if you need a little more review. Now, how is DNA actually copied? You should be able to draw for the test this specific picture which shows all the different enzymes and proteins which are involved in the DNA copy process and all the steps which are involved in this. Let's talk about that in a little more detail. First of all, DNA is copied during the interphase, during the S phase of the cell cycle in the nucleus of eukaryotes. And in prokaryotes, that just happens in the cytoplasm along the nucleoid region. After the S phase, the DNA will be doubled, and if coiled, will form that X-shaped chromosome that we're familiar with. Now, DNA will actually, the first thing that will happen is that it will uncoil itself up in what is called a replication fork. The DNA will basically unzip, and so each of these strands can be completed semi-conservatively to form a new copies of the DNA. When you actually look at the copy process of eukaryotes, though, you see that it actually happens all the time in several strands all at once. It's almost like you have multiple areas of the DNA being copied all at once. You don't have just one replication form traveling. You actually have many of them all over the DNA structure. And that makes the copy a lot faster and more efficient. And we call each of these regions of DNA copy a replication bubble, which is led up by a replication fork, which then closes back up after the replication is completed. And in case of eukaryotes, you're going to have multiple of this, while prokaryotes will only have one replication fork at a time, traveling through their circular chromosome, while eukaryotes will have hundreds of these per chromosome and several chromosomes doing it all at the same time, like you see in the picture here. DNA is copied semi-conservatively. No matter which strand is being copied, you're always going to have the basic same thing as a starting point. You're going to have topoisomerase coming in and coiling, untwisting, and stabilizing the molecule to stay that way, because the DNA likes to stay paired up. So that enzyme will stay there to stabilize the structure and to actually uncoil the twist of the double helix and make it stay as a strand that's not twisted. Then comes in helicase, and what helicase does is it unzips the genes and actually makes the DNA into the Y fork. The fork doesn't like to be that way though, so it's necessary to put single binding proteins to stabilize the unpaired strands and prohibit them from actually zipping back up. Then you come in with primase, which is an enzyme that comes in to mark the starting spot for the synthesis process by laying down primers made of RNA, which will eventually be replaced by the building blocks for the nucleotides of DNA-like nucleotides by a different enzyme called DNA polymerase. In eukaryotes, the DNA polymerase that does this is DNA polymerase type 3. There's different types of DNA polymerase. And the way it does it is that it gets a nucleoside triphosphate, which is a charged nucleotide molecule, which has two extra phosphate groups, and it lines up with the template strand by making sure that base pairing rules are correct. So if it's an A, it will pair up with a T. Then it will cleave the energy that's stored in those extra phosphate groups, and using that energy it would actually create the bond that will pair the strands together. And that's kind of how the DNA process will happen, in regardless of the strand that you're looking at. But there's a problem in the DNA. One strand is going to be copied continuously, and that's called the leading strand. And the other strand is going to be copying discontinuously, and it's called the lagging strand. The whole thing is that DNA can only be copied in a 5 to 3 direction because it can only be read in a 3 to 5 direction by DNA polymerase, which means the strand will be formed in a 5 to 3 direction regardless of what strand you're talking about. So in the leading strand, that's not a problem. You're going to start far away from the replication fork in what is called the origin of replication, somewhere down here, and you're going to copy continuously in this direction. But on the lagging strand, you have a problem. DNA polymerase is too big to fit in the replication fork, and so it cannot start copying from here, which is where the 3 direction is, since the DNA strands are anti-parallel. Which means it's going to have to copy in the opposite direction towards the point of origin of replication, and we'll have to copy in fragments. Since it won't fit in the actual Y fork, it will have to start copying away from the Y fork. So the primers will be laid somewhere here, and they will copy in this direction. Let's see how that actually works strand by strand. So again, the leading strand will copy it continuously from the point of origin towards the Y fork with no problem at all. That's how the leading strand will basically be copied. But on the lagging strand, because the DNA is anti-parallel, you have to go in the opposite direction. The DNA RNA primer will be laid up away from the Y fork, and DNA polymerase 3 will copy the nucleotides on that region first. Then it will jump to the beginning of the strand after it has unzipped a little bit more, and a new fragment will be made. And then DNA polymerase 1 will come in to complete and connect the fragments, and then ligase will come in to actually stitch the backbone and make it in one continuous molecule again. And that is why you need an extra enzymes, because of those Okazaki fragments. So the reason why they exist is because DNA polymerase 3 will not fit in the replication fork, and so that means it needs to copy the lagging strand in pieces. And then DNA polymerase 1 is going to be necessary to complete the pieces, and also to ligase to actually stitch the pieces back together. These are the most important enzymes which are part of the DNA replication process. If you're missing one of the enzymes, things won't happen the way they're supposed to. Without topoisomerase, nothing happens because DNA will not uncoil itself. Without helicase, it will unzip itself. Without single binding proteins, it will coil itself back up. Without primase, polymerase will not know where to start. Without polymerase, no copy gets made. Without ligase, the backbone of the Okazaki fragments will not be stitched together into one continuous strand in the lagging strand of the DNA molecule. Each one of these enzymes is fundamental to the copy process. After DNA does all of this, it sometimes makes mistakes. DNA polymerase is not perfect. Sometimes it lays down the bricks the wrong way. Sometimes it pairs A with C or with G. And that will cause the DNA molecule sometimes to have an abnormal shape or be longer than it should at that point, as you can see, because it will actually have 4 rings in the middle or just 2 rings in the middle, or maybe even it will have 3 rings in the middle but will be unpaired incorrectly. And so you have a different kind of enzymes, which we'll call proofreading enzymes, which will come through and actually double check the DNA to make sure there's no mistakes like that. If there are mistakes, a different set of enzyme called excision repair enzymes will come through, cut the bad piece, call in DNA polymerase to repair the problem, and then ligase will stitch the backbone back up and complete and correct the problem. Before this, DNA makes one mistake every 10,000 bases. But after the switch repair enzymes actually correct the problem, the DNA only makes 1 in 10 billion mistakes. And it's still not perfect though. Why would it purposely leave those mutations behind? Would life have done better? Well, maybe, perhaps. But the thing is that these mistakes are actually important. If it wasn't for these mistakes, you would never change. We'll all be clones, and we would never evolve. And since the environment is changing, we would eventually go extinct. And because we will all be the same, there will be less diversity, and we will be more likely to actually die because of one thing. If one thing kills one of us, it will probably kill all of us. And that's the problem with not having mutations. So life left them on purpose. There's also a problem with the end of the DNA molecule. You see, when the leading strand is copied, there's no problem. It's copied from the very beginning of the origin of replication. But on the lagging strand, because you're going to have to lay down the RNA primer on that last fragment, that little piece is not going to get copied. And so the problem is going to be that every time you make a DNA copy process, you're going to have shorter and shorter ends to the molecule. And so you're going to have the DNA shortening over time. And so if you have a circular chromosome like a prokaryotes, you don't have this problem. But if you have a linear chromosome like you have in eukaryotes, you're going to need to repair this problem. And so what telomerase does is a different enzyme is going to come through and add a random amount of nucleotides at the very end of the chromosomes and create what is called a telomere. And this telomere will prevent the cell from losing DNA after every single copy process. And if you didn't have this, what will happen is that you will get older faster because the DNA will lose its integrity and cause non-mutations. And that's why some cells, after many generations, since telomerase is not exactly perfect, will actually become older and senescent and basically become useless cells because they're losing actual DNA once the telomeres are completely eaten up. Imagine what would happen though if you didn't have telomeres altogether. You would basically, after every generation, the DNA would get shorter and shorter because during the formation of gametes, the same thing would actually happen. That's actually a good thing for cancer because since they replicate so fast, it's more likely that they will actually lose telomeres faster. So cancer cells would actually spread even faster if it wasn't for telomeres. Interestingly enough, though, some of the most aggressive kinds of cancer have cells which never lose their telomeres. So that's something going on there that we could possibly research to actually make sure that our cells never become senescent and we can get eternal life. But that's never happening. Life left a mistake there on purpose to help us age so that the new generations could replace the old generations and evolution could go on. The last thing we have to talk about is the idea that there are differences between prokaryotic and eukaryotic cell division. Basically, in eukaryotes, the division process will take longer because it's more complicated, it involves more enzymes, it's more complex, it will happen slower, and the Okazaki fragments will be shorter to manage all the problems that could come up because if the Okazaki fragments were longer. And this all has to do with the fact that eukaryotes have multiple linear coiled chromosomes, while prokaryotes have singular, circular, naked chromosomes, which don't really have all the histone scaffolding or any of the problems that you have. Alright? And that's DNA replication, and I hope you learn a lot and don't do anything that would make your mama proud.